Hopefully you all heard recording in progress. I certainly heard recording in progress, but hopefully you also saw a little thing that says, hey, you're about to be recorded. And then you just say, yes, you agree. And if you don't want any record of your voice to be made here, then you simply use the chat in the Q&A if you want to say anything. But we'll get started now. So my name is Mirka Zaplatel, and I'm the Director of Education at the McAuliffe-Shepard Discovery Center. I want to thank you all for joining us tonight. This is now, actually, we are now at just over a year of doing this virtually. So we started last June with our first virtual version of Superstellar Friday. And so now we're back to June again. And I don't know if we imagined that that would be the way things go, but we're planning on doing them virtually at least for a little bit longer. So we're so happy that you can join us this way. And someday it would be great to actually see people in person again. Tonight, I'm very excited because we're going to be getting into something that I think I don't necessarily understand very well, but I think lots of other people don't always understand very well. And that's taxonomy and figuring out that there is a new species and what that means and how you figure that out. And so Dr. Cate Craig is an expert in a number of things, including hermit crabs and taxonomy. And when I talk to her, she tends to say that she thinks of herself as a taxonomist who happens to work with hermit crabs. So that's why I thought, well, this was a great opportunity for us to learn something about, a topic that we probably don't encounter very much, but certainly when there is a new species in the news, it's big news. So I think this gives us hopefully a new window. The way that we're going to be doing this, as usual, is she will be sharing her screen and making her presentation, and I will be monitoring the chat in the Q&A. So you can make your comments, ask your questions, and then periodically she will stop and ask if there are any questions. And if there are, I will feed them to her. And if you ask a question and it takes a little while for me to get to it, don't worry, I promise I will. Sometimes I'm trying to group questions together or maybe put things in a certain sequence, but I promise you will get to all of those. And so now what I'm going to do is I'm going to mute myself and turn off my video and turn it over to her.
Hi, everybody. Hi, everybody. I'm Cate Craig. I'm just today going to introduce a couple of things to you about how species get described, because there is a process by which we decide what's a new species and some methods that I think a lot of people aren't aware of. So I'm going to go ahead and start us with a video. Maybe. Uh-oh, hang on.
A team of scientists with the National Oceanic and Atmospheric Administration, or NOAA, have confirmed the discovery of a new gelatinous marine animal just off the coast of Puerto Rico. The species of ctenophore was first seen during a remote underwater expedition in 2015. They are also known as comb jellies. The researchers described the creature as resembling a hot air balloon that moves across the sea floor. NOAA says it's the first time scientists have identified a new species using only high-definition video.
All right, so hopefully we're back from the video. And the reason I chose that video is just because it shows you an example of the mainstream news promoting a new species. That was described by scientists, and they say described. And so today I'm going to talk a little bit about what that means to describe a species, because it's a little more involved than just CBS picking up sort of a conversation about what you have seen as a scientist, right? There's actually a process. There's a process by which other scientists sort of approve this species. And that's what I want to talk about today, because I think that is a unique perspective that maybe isn't a common one that's talked about when we talk about new species. So this slide right here shows you the title of my dissertation. So dissertation is just a big long paper you write and then they give you a degree. It actually takes longer than that and it's hard. But this is the title of mine. And you can see here, here at the end, descriptions of 2 new species. So in my dissertation, I described 2 new species of hermit crab, and I'm going to talk to you a little bit about the methods by which I did that. The objectives of my dissertation were a little bit involved. So one of them was to develop a stable taxonomy, and taxonomy is what I'm going to talk about today. There we go. Taxonomy is what I'm going to talk about today in a little bit more detail. Over here, we don't care so much about these things today. We don't care so much about evolution of characters. What we care about is talking about how taxonomy contributes to biodiversity estimates, and then how molecular techniques and morphology, so the physical structure of an organism, comes into play when we talk about describing new species. So when we talk about a species, we're talking about several things. We're talking about sort of classification, and one of the earliest classifications that was developed started with Aristotle. And Aristotle took this idea, all these animals that have been discovered in culture, and he divided them into 2 basic divisions, some with red blood cells, or some with red blood and some without red blood. It turns out, in terms of evolution, he was pretty spot on, right? So we don't see a lot of sort of spiders being closely related to fishes, right, as we go forward with our study of evolution. So he had that right. He was a little bit wrong about crustaceans. There actually are some crustaceans that have red blood, very few of them. But he was really one of the first people to start organizing these animals. Next, we had Linnaeus, and Carolus Linnaeus was Swedish. He decided we're going to get organized. We're going to take these animals that we know about, that we've discovered over time, and we're going to start to organize them. And the sort of school of thought he promoted was one called taxonomy. And taxonomy is just the way that we group these and the way that we name them. And Linnaeus's sort of concept was this binomial nomenclature. So bi is for 2, and nominum is name. All right, so that's a Latin word for name. So he's saying 2 names, and those 2-part names are written genus and species. So the grouping of genus is just a group of species. So we're kind of working our way up this tree from small to large, where we start with species, and then as we go up this little mushroom, right, we get larger and larger groupings. So genus is many species, a family is many genera, so that's just plural for genus. Then you have orders that are many families, and so on as you go up the sort of progression. Carolus Linnaeus used to say, if you do not know the name of things, the knowledge of them is lost too. And in fact, in some translations of this phrase, sometimes he says the knowledge of them perishes. So I think that's I don't know, pretty strong language, but I think it really captures sort of how taxonomists feel about naming and classification. So I worked on hermit crabs, and hermit crabs are really interesting. They're very diverse in terms of their shape and their body shape and how they're organized. This is a king crab. A king crab is actually a hermit crab, and there's some aspects of its physical structure that lead to that, and we can talk about that later if you want to. But mostly they have these soft abdomens, and they live in snail shells for the most part. When we talk about hermit crabs, we're talking about a superfamily that has 7 families and about 1,000 species. It's actually closer to 1,200, but it's kind of splitting hairs, so let's not worry about that. Then we have a family inside that superfamily with 22 genera. And here I've got some of those genera listed out for you, and then the number of species that's inside of them. So that gives you an idea of kind of the progression of classification that we're working up to. So when you're looking for a new species, or when you think there might be one, you're beginning with a suspicion. All right, so it's important as a scientist to be very suspicious. A nicer way of saying that is curious. So you can start with curiosity as well. But for me, it always comes up as I look at these animals or these groups of animals. There are little hints, there are little physical features or maybe habitats that they live in that make me suspicious that maybe they're different than the others. Darwin has some classic examples of that, one of them being the finches. So he looked at all these groups of birds that he found on his cruises in the Beagle when he did a lot of his early work on natural selection. He noticed something different about groups of individuals. So not about a single bird, but about a group of birds that hung out together. And he noticed differences in physical features. So you can see here, this is a drawing of their different beaks. But he also noticed kind of, if you look down here in this chart, this wheel, it shows you that they all had different eating habits, all right, that were related to their bill shape. And he started to think in his tree of life, he started to think that maybe there was some system by which these animals were organized in terms of their evolutionary history. So when we're looking at species, we use what's called a species concept in order to say, okay, how are we going to determine this is a species or not, or it's a different species from another? This table is just showing you the diversity of species concepts out there. So you can see here on the left, we've got a bunch of different species concepts. Here we've got a bunch of descriptions of them, all right? And they're really quite diverse, and depending on who you talk to, some are better than others. So which is best? Well, to me, the species concept you use is dependent on the type of data you're able to gather. So do you have a physical animal? So if we remember that CBS video clip, they didn't catch the animal, they just had video footage, right? So in that case, they're going to use a different species concept than they would use if they had been able to collect a genetic sequence from them, right? Ecological or conservation importance can be important to the species. So if we're looking at, does it have a unique ecological niche, right? That can be a clue. And then requirements of various funding and regulatory bodies. So things like the Endangered Species Act, for example, some federal protections of wildlife, they require certain criteria to be met in order for them to define it as a species. So for example, maybe they require a certain amount of genetic divergence in a certain gene. So COX-1, for example, needs to be, a certain gene needs to be 10% different among or between two groups in order for that second group to be a species. All right, so that's some of the examples of data you might be able to gather. One of the most commonly cited species concepts is the biological species concept. This concept is where you have two groups of animals, they have natural interbreeding, and then they produce viable and fertile offspring. Then we say they're the same species. So if they can't do that, then we say they're different species under the biological concept. The morphological concept is more like what the scientists used from the CBS video. So they're looking at that video and they're saying there's unique physical features. So morphology is just the shape or structure of an organism. So we're saying in that case, they had unique physical features. And then evolutionary species concept is one you see a lot in like paleontology, right? So talking about a unique evolutionary role and fate. Like if we see all of a certain group of animals disappearing in a certain layer of the stone, then we say, okay, well, those groups of animals may have been the same species. The concept I work with the most is the phylogenetic concept. And the phylogenetic concept has a bunch of different flavors, but the flavor I talk about a lot is monophyly. So when I'm talking about monophyly, I'm talking about a group of species that are all related, that share the same common ancestor. So if you think about something like your siblings, let's say you have, I don't know, how about 10 sisters? So let's say you have 10 sisters, you have a common ancestor in your father. So your family, your sister group is monophyletic, right? So when we're talking about these species, that's what we're looking for when we're looking for monophyly. There's a lot of different methods for determining monophyly. And again, that's related to the type of information you have, right? So if you have genetic information, then you're going to use a different method than if you just have morphology, if you just have, say, claw shape or beak shape or something like that. And in modern sort of analyses, we consider all those types of data, so genetics and morphology. And one of the ways we do that with our genetic data, if you see over here on the right bottom, if we pretend that these A, C, Gs, and Ts are representing a gene sequence, then we can look and we can say, okay, well, how is this top sequence and this bottom sequence, how are they different? And if we look out here at our individual species, how have they changed between each other? All right, and then we know for this gene, okay, well, this animal right here has a different sequence than this animal down here. They're different. All right, and we can build these sort of family trees of species. This is a bigger version of one of those. I know it looks a little scary just because there's a lot of lines and a lot of arrows and things like that. But what we need to worry about when we look at this figure is not necessarily the names or the types of animals or anything. We want to look at the shape of the tree. So if we're looking for something that's monophyletic, we're looking for something that has the same branch here at the base. So all of these little tips, we want them to be connected at the same branch, and that's monophyly. So you can see here we've got these amphibians, these frogs, salamanders, and caecilians, which caecilians are just this really weird kind of sort of semi-fish, semi-salamander kind of thing. They're really weird. But we can see that together they share a common ancestor. They're monophyletic. So that's what I'm looking for when I look for separate species. So what we're looking for when we're looking for these animals is we're looking to diagnose something. So I've highlighted that for you. Diagnosis. So just like a disease, we're looking for certain symptoms essentially. So we're looking for certain symptoms that make this crab different from this crab over here on the right. Right. So when we're talking about this species, so genus, Areopagoristus, species, Hewitti, we can see that we've got sort of various characteristics that make it unique from this one, which this should be Areopagoristus hummi, but we'll see him again. So don't worry too much about the names. But this animal has blue eyes and is less hairy. So we ask ourselves, well, are they different enough to qualify? And the method I use is called integrative taxonomy. And integrative taxonomy is using those multiple lines of data to determine whether this is a species or a genus. And that's things like morphology, genetics, or even geography. All right, so maybe their geographic distributions don't match, or maybe they live at different depths, right? That can be additional evidence that maybe they're different species. So this is a paper we published that used a lot of that different types of data. So this went to Zootaxa, which is a scholarly journal, which is basically like a kind of a boring magazine for scientists. I mean, we don't think it's boring, but it's not exactly like, you know, People or Cosmo. All right. So we've got our Zootaxa, this magazine, and you can see here I've highlighted the types of data we used. So we used gene sequence data from these specific genes. We used morphology. We used a phylogenetic approach. So we used that concept of evolutionary relatedness. We used geographic distribution. So we said one of our species is from the Gulf, and one of them is from close to Suriname. And then we used ecological data. So we said, okay, well, some of them live in deep water, some of them live in shallow water. Now we have to actually describe the species. So we've decided that they're different. Now we have to describe them like literally, like you sit there with the animal under a microscope, because most of these crabs are millimeters in length. All right, their carapace is millimeters, like 2 or 4, right? And you look at the morphology and you write down exactly what you see, like exactly what you see painstakingly. And you use the lingo, right? So you got to use the lingo. Like, you don't want to be talking about this crab's leg, right? Its technical name is a paraeopod, all right? You don't want to talk about the hairs on its leg. You want to talk about the setae on its paraeopod. And those features are called characters, right? And we define our groups by multiple characters. So it's not enough for a new species just to have one different thing. It has to have multiple different things. All right, just one thing is something that makes taxonomists suspicious, right? Again, with the suspicion, right? Makes us suspicious that we don't really have new species. So what does this really long drawn out process do? Well, it provides evidence that you looked, right? If you write down pages, because right here, this picture, this goes on for pages, all right? You write down all this detailed information, it proves to other people that you looked, right? So it says, you know, I looked hard and I'm pretty sure it's different. It also establishes your street cred. So if you don't do this, other taxonomists question whether you're good at your job, basically. So you want to do this very detailed, very accurately, and then you get to become the authority on that species. And I'll show you what that means right towards the end. One of the dangers here is synonymies. So synonymous just means that two species are actually the same. So you've described something as new when it was actually the same as something someone else already described. All right, so that is a danger you want to avoid, and you avoid it by reading these very detailed descriptions, which is very time consuming and not always everybody's favorite thing. So here's an example of one that we described in the dissertation. We've got what we're calling near Tortugae, and we've got Tortugae. So Tortugae is just its species name. Pagoristus is its genus name. So these are of the same genus. They're very, very similar. And they're very similar. In fact, one of the few things we could find that's different is their nose, their rostrum. So they've got this little point that sticks out in between their eyes. Sticks out in between their eye stalks right here. And it's a little bit different shape. So I drew it here. I drew one that's very pointy, one that's kind of rounded. Also, as you can see in the slide, they're very different colors. So that's something that we could use to identify them. So here we see these two groups of animals that are different. I question whether they're the same species. So I question whether they're the same species. I did notice some morphological differences, and this is the integrative part. So now I'm going to mix that morphological data with genetic data, right? So if we look and think back to our phylogenetic concept, we used our genes to generate a family tree of species, all right? And you can see that here, this sort of, it's a little more confusing just because all the branches aren't the same length. But I want you to look up here in this orange part, all right? And you can see where this 100 is, it's coming in on a single branch. And then you can see it splits, and now we have all these light orange Pagoristus tortugae, and we have all these dark orange Pagoristus near Tortugae. So that means because we use genes to do this analysis, that means the genes of these dark orange animals are different than the genes of these light orange animals. And that helps support the idea that this crab over here is a new species. So that plus the morphology gives me a really good case, a really good sort of case to argue that this is a new species. More information is always better. We saw it with another one too. So here's another guy, and you can see again, we get that same trend between the blue and the light blue where the genes are different. In this case, we have another sort of ace in the hole. In this case, the geographic distribution of the dark blue and the light blue isn't the same. So our dark blue animals come from Central America only. So far, we may find more in another place, and that'll confuse the issue somewhat. But right now, we only know of them from Central America. Okay, so now we've described it. When we described it, we had to pick something called a type. And the specimen that we pick is called the holotype. The holotype is a specimen by which this species is defined. So an actual physical animal that anybody who questions your species will need to go back to and look at and confirm whether or not the species they're describing is the same. So it's necessary for comparison of future animals. The problem with the holotype is that we can lose it or we can destroy it. And that's what happened to these animals over here. So this old drawing on the left is of the animal that is the type for one of my main genera I work on. And we aren't even sure where it went, but we think it got destroyed in the Chicago fire. We're pretty sure. And right now I'm trying to prove that this animal over here on the right is the same as this one on the left, but I don't have the type, I don't have the holotype that was drawn here on the left, right? So that is a problem with the holotype. You got to keep track of them. Lately, when we describe a type, we're expecting photographs and DNA sequences as part of the description. So that will help sort of combat the problems that arise when we lose the type. So, and then when you have the type, you have a type locality, and the locality is the place where the type was found. So the physical place where if I had to go get a new one, I could find it, theoretically, right? So that's kind of, it's a nice idea, but there's no guarantee it would be the same. But if it came from the same place, it's probably the same. The problem with this scenario over here on the right is that we don't know exactly where this left animal, this old tiny animal, we aren't exactly sure where it came from. The notes from the person who found it say it came from Chile, which Chile is really big. It has a very long coast, so we don't really know. So key in replacing this specimen would be the locality, and this one's a little bit of a mystery so far. So now we've described, as we described, we did something called illustration. So illustration is just drawing. It's kind of old-timey. So this is a really old school thing to do. It's really important though. There's a lot of discussion right now about whether or not we should just use photographs, okay? And there's some problems with photographs just because of sort of how they need to be taken in order to get enough detail on the animal. But illustration, I think, is important, more important, because it shows what your eye would actually notice. All right, so if we look down here on the right, you can see some illustrations that we did to go with one of our earlier papers. And you can see that they're basically reduced to very sort of detailed, but simple line drawings that if you were looking through a microscope or looking at an animal in your hand, you would be able to kind of see those features on the crab. So that's why illustration is so important, is because it shows you what you're actually going to see, not sort of additional detail that your mind would filter out. It's also inexpensive. It requires minimal space and equipment. So depending on the animals you work on, you may not even need a microscope. It's also fairly quick. So I could go from a crab to an illustration like this in a day. And it's predictable. You know that you're going to be able to do it. You know that someone who is reasonably well trained is going to be able to do it. And we have a long legacy of it, so it's easy to compare to older work. Here's a camera lucida, and I'll show you that on our next slide. This microscope right here in the center is kind of what we use. It's really similar to the one I wrote my dissertation with. And then this black thing that sticks off to the side, that's a camera lucida. And the camera lucida is basically able to take an image of a specimen you're seeing, pass it through a mirror or pass it up against a mirror and project it onto the surface that you can draw on. Here's how that works. So down here at the bottom, you've got light. It's passing through an object up here. And then that light is coming into the prism in the center of the microscope. It's bouncing out to a mirror, and that mirror then projects it down onto the drawing surface. So what you see is you actually see essentially an image of what you're seeing under the microscope, and you can just trace over the top of it. There's a little more to it than that. I mean, you got to really be practiced at it, but you actually see the animal exactly as you see it with the microscope. And so you're getting a replica that's not sort of artistic or stylized. This is my space that I worked in during my dissertation. You can see here's my microscope. Here's my camera lucida off to the side. Here's all my crabs. So we store our specimens in ethanol, really high proof ethanol. And that helps preserve their tissues so that we can extract DNA later if we want to. It helps preserve their sort of hard parts, their calcified parts, for later morphological evaluation. So ethanol is a pretty good preservative. still, if it's older than about 10 years, it's really difficult to get DNA out of with the methods that we're able to use. Paleontologists who extract DNA, they have some special tricks, but we aren't able to use them on our material. So about 10 years is our window for being able to extract. So that's where I worked in the lab during my dissertation. Here's where I work in the lab now. So since COVID and since my advisor retired and they shut down our lab, now I have my microscope here. There's my camera lucida still. And then I have this other important machine for the study of fabric, and then some additional specimens here. Many colors are preserved, and then the rest of my specimens down here. So there's a modern twist on illustration that's very exciting, although it has some pitfalls. That modern twist is CT scanning. So CT scanning is just basically like a circular X-ray that is able to produce these very accurate 3D images. And my friend Janice does these. He's very, very behind them in terms of them being kind of the wave of the future. I have some doubts, but they're good for rare specimens. So things that we don't have very many of or that we find very rarely. And that's partially because they're non-destructive. So it means that I don't have to rip off all the animal's parts in order to get a good view of them, in order to draw them. All right, it means that we can leave the animal relatively intact. We can put a coating on it, put it in the CT scanner and get this beautiful 3D image. Now, these images only really capture the well calcified parts, so the very highly crusty parts. And that's not the whole part of a crab, right? Crabs, they have some softer parts as well. I question whether or not they're almost too detailed. So if we look over here on the left, you can see here's all these setae, all these hairs, right? And the CT scan has caught them so well that you almost can't see through them. With your eye under a microscope, you would be able to focus up and down and focus through those to get a view of the claw. Versus here, he's had to like manually essentially remove them through manipulation of the image. So I think there's cases where they can be too detailed. It's also time consuming to do, and there are limited resources to do it with. So it's expensive, it takes a long time, and not everybody has a CT scanner. But pretty much most scientists that work at universities or museums can come up with a microscope and a camera lucida. Okay, now we've decided it's new, we've described its physical features and drawn it. Now we need to name it. And the naming thing is, there's rules. So you can't just name it whatever you want. You are not allowed to name it after yourself. Okay, so you can't go around naming a bunch of stuff, after yourself. The governing body is the International Commission of Zoological Nomenclature. So zoology, just the study of animals, right? And they wrote the International Code of Zoological Nomenclature. Right, here's this book. And this book it's all the rules, right? And we have to have rules because there are thousands of species described every year of all kinds of animal. And so we got to keep everything kind of standardized, or else things get named the same thing, or things get named things that don't make sense. All right. And then you get into name battles where, for example, you name a genus, but there's a genus of beetles that's already named that. All right. The International Commission of Zoological Nomenclature takes care of that. They follow up on that and they're like, nope, you got to change your name. The other thing they look at is your syntax correct? So remember, these are Latin names. And what that means is that they have to be conjugated a certain way. All right, so they have to be a certain way when they're plural. They have to be a certain way when they're female. They have to be a certain way when they're male. And so the ICZN deals with all that and makes sure that everything is kind of consistent and put together. So there are rules. So what's in a name? Why do we choose the names we choose? Because as the describer of the animal, you do get to choose. And the ICZN just follows up and makes sure it's legal, right? So why do we choose them? Well, the first reason we choose them is we combine Latin roots and prefixes to include useful information. So Diogenes was that family of hermit crabs that I work on. Diogenes was a hermit in Greek history who was known for living on the streets in a jar. All right, so he was a hermit. These are hermit crabs. He lived in a jar. Hermit crabs lived in a shell. So it's kind of apropos, right? It's kind of makes sense. It helps you remember kind of the nature of the crab. So that's important. Pagoristus tortugae, which we saw earlier, tortugae, its type locality, so the place that it's defined by is in the dry Tortugas, right? So Pagoristus tortugae. This one is really cool. I picked it because it has so many pieces of information in it to kind of describe. So Parapagurus pilosomanus. It just seems like a really weird word to say and spell, but it actually is a super easy way to remember this animal. This is the animal over here on the right. And so para means that it's alike or somewhat like. Pagurus is another genus of hermit crabs, and that genus happens to have large right claws. All right, so it's like Pagurus. It has a large right claw. Pelosi refers to pelosity, which is hairy. All right, so we see Pelosi, pelosity, hairy. So now we've got it's somewhat like Pagurus and it's hairy, and then manus, the manus is the hand or claw. So it's somewhat like Pagurus with a hairy claw. All right, so we include useful information in that. And you can see over here, it's a little difficult in this photo, but he has hair on the margin of his claw. So that's where he gets his name. Other reasons that we choose the name we choose can be kind of, I don't know, silly or sentimental. We do it to be nice to our friends. So Areopagoristus is our genus. Lemaitre is our species. This is named for a guy named Raphael Lemaitre, who's a friend of mine. And he is male. That means it has to end EI because of how Latin works, right? So Lemaitre. Areopagoristus McLaughlinae is named for Patsy McLaughlin, who is a very famous hermit crab worker, and she's female and it ends in AE. The other thing we do is we name things in ways because we think we're funny. All right, so here down here, you've got Hairyplax severus. This crab right here. Harry is one of the people who found it, who collected it. His name is Harry. And then because he thought it was funny, the person who described this then named the species Severus. So very witty. There we go. And then Synalpheus pink floydii, at least this makes sense because his claw is pink. All right, so Synalpheus is the genus, existing genus, and then pink floydii with a pink claw. And I happen to know the people who named this one did this over beer. So they were out and about and decided pink floydii was the way to go. All right, now you publish. So now you write it up, you send it to a science magazine, boring science magazine. So not a fun one like Science or Nature, but taxonomy papers usually go in ones with other taxonomy papers. So, and now you become the authority. And the authority follows the species name. So here we've got Pagoristus tortugae again as an example from the dry Tortugas, was described by Schmidt in 1933. Phymochirus tunneli was described by Felder, who's my boss, et al., which et al. Just means and others, in 2019. So this paper right here, where we described this Phymochirus, had 3 authors. So once it's got 3 authors, you have to just say and others. And then the year. So once you've published this, you get to, every time someone uses this species name in literature, they have to write after it, Schmidt, 1933, or Zapletel, 2021, or whoever the describer is, and then the year. Now you have to spend the rest of your life defending your decision. All right, so your paper goes through peer review. And other scientists look at it, they say, yeah, they did the right steps and we agree with them, and we're going to publish this paper. And then the passage of time kind of cements this species, right? Because many people over a long period of time will look at that Pagoristus tortugae and say, yes, it agrees, we agree, everybody agrees. until someday, hundreds of years in the future, someone looks at it and says, I disagree, and overturns your species, and you're very sad about it. But nobody knows that because you're not around. So larger groupings, the genus. When you're looking for a genus, you have to group your species. And when you group your species, you're not looking anymore about this species diagnosis, the things that make these two different. You're not looking for that anymore because you know that. Now you want to go up here to genus, a grouping of species. Then you look for something that they all share, right? In the case of these crabs, they all share a certain number of gills. So crabs have gills, they're in here, right? And so they all share the same number of gills. And so we know they're a genus together, multiple species. So there's a lot of different people working in taxonomy. These people happen to be crab taxonomists just because I am a crab taxonomist. That's the people I know. Here, right here in this upper corner are Dr. Felder, my boss, and Raphael Lemaitre, who that crab is named from. There's also shrimps named after Felder, Felderi, and Raphaeli. Right? This lady right here, she's very nice. Her name is Dwilis Uraheu. She's Indonesian and she's very famous. This nice little lady right there, and she's got her camera lucida and her scope. This is JC. JC is responsible for Hairyplax severus. So when you think about that, think about JC. He's from the Philippines. He works in Singapore with this gentleman over here, Peter Ung, who is also very famous. We're all famous in our own way. It's just we're not movie stars. We're just famous in crabland, I guess. This is Janice, who does the CT scans. He is German. He works out of South Africa. My lab mate, Brent, he's at Jackson State University. And then here's me. I'm on the back of a ship that we use in Louisiana to go out to the Gulf of Mexico and collect crabs so that we can describe more species and get a better understanding of how biodiversity works in the ocean, kind of what's down there, where it is, what it is. And keep track of these kind of things just because, I don't know, it's our job. So yeah, so if you guys have any questions about like taxonomy methods or hermit crabs or species concepts or anything like that, I think that would be a great place to go next. And I'm going to go ahead and close out of this and stop sharing my screen, I think. And then Mirka can come back and be the boss of us again.
Okay, thank you very much. So if you have any questions, please put them in the chat or the Q&A. I have a question. So you gave this scenario of, so you describe a species, but now 100 years from now or something like that, somebody says, oh, I disagree. I would imagine it's a complicated process, but is it, how complicated is it to say this original species from 100 years ago is in fact not correct?
It's not complicated per se. You need a lot of evidence. So I'm in this situation right now on a couple of species where it's like I've looked at them and I've looked at the, again, you have to go back to the type. Right, so if I have a crab in my hand that I collected yesterday and I think it might be species X, I need to go back to the type specimen of species X and compare them. And when I do that, if I look at that and I say, you know, this isn't what you described in your paper in 1894, then I have to write that up and I have to send it to the journal and other people have to read it and then they have to argue about it. And so there is a process, but basically, honestly, it's the kind of thing where the longer it's been, the more support you need, right? Because the more accepted it is.
So does it ever happen, just because I know that it'll take a while sometimes for people to do their work and then to get it published, does it ever happen that you would have two people who find the same thing, describe it, but obviously they don't know about each other. And so then how do you deal with that kind of situation?
Yeah, so that's actually, if you look at older literature, a huge problem, right? Because they used to be like sending books to each other on sailboats, right? Or traveling around to different museums, and they were at different museums at different times, and you know, that is really common. It's still common today, just because we're all working at different speeds in different places. And when you describe a new species, you are responsible for making sure that there's nothing else that's already described that's like it. But you don't catch it all, and someone will catch it later usually. And so in every description you write, there's a whole section for that, for synonymies, for saying, okay, well, I'm describing this, and the one that Schmidt described in 1935 is the same as this, and the one that Wass described in 1935 is the same as this. And so you document it, but you have to go and do the due diligence of going to the museum and looking at the animals and that whole thing, if you're gonna try to synonymize things.
Do people ever get in really big fights about this? Arguments about who was first, or
Yeah, I'm in one right now.
should
I'm in a synonymy battle right now, and it has to do with that genus that I work on, Pagoristas, where the type specimen has been lost, and the type species was synonymized with another species. So the old species that was the type species was joined to another one later in the 1950s. So this original animal's from the 1850s. It was blended with this other animal in the 1950s. And now someone today wants to split them apart again. He says, no, they're not the same. And I say, yeah, they are. So we're fighting.
Okay, so is there a judge and jury for this? Like, how does it get? I assume it plays out in the literature and people reviewing it.
Yeah, it plays out in the literature and people reviewing the literature and people writing reviews on the literature. So I have reasons that I think this, they have reasons that they think this, and it's up to the scientific community to decide whose reasons are the best reasons. So we're just kind of putting it all out there and waiting for everyone to chime in.
Have an opinion.
Yeah, oh yeah, they all do.
So we have a question. How do you describe an animal that has only been seen via camera, like the new ctenophore species from the video? So how does that work as being a species?
So that would be like the purest form of morphological concept, right? So all you have is what you can see. And ctenophores in particular are defined by the number of rows of combs. And those combs are on the outside of the animal. So because they're jelly creatures, a lot of their features are readily visible. So I would think in the case of the ctenophore that was described based on video, that would be very conducive to that. A lot of other animals, I just don't think you could based on video alone. You would have to have some other physiology or habitat or something. I mean, the habitat helped them out too, right? Because it's the only thing they've found down there that's that deep water of a ctenophore. But yeah, I think from video you can for certain types of animals, if you can see enough morphology to be confident. But remember, anytime you do this, anytime you describe a species, you are putting yourself out there ready for disagreement in 20, 30, 40 years, to stand there with egg on your face and say, I was wrong, I jumped the gun, I didn't check carefully enough. Right? So you got to be pretty confident to put it out there, especially on us, you know?
Yeah.
So.
Okay, so I wanted to ask a question, I think that kind of connects maybe back to something that you alluded to begin with, which was, you know, what does this help us with in terms of diversity and understanding, like why ultimately is it important that we know that these two were actually two different species, or they weren't two different species?
So I have kind of I have a two-pronged sort of approach to that sort of question. The first one is that, you know, if we go back to Socratic principles and Linnaeus, like, you know, in nature, there is something wonderful, right? And so knowing about the things that are there is kind of our sort of our calling as biologists, as naturalists, right? So there's that aspect that makes me have the warm fuzzies, right? But also, I think when we're talking about conservation, it's really important, but not in the ways that a lot of people talk about it. Like a lot of people are like, oh, we have to save as many species as we can. We have to, you know, but to me, biodiversity is really important because we have to preserve the habitat. We have to know which habitat is where the diversity is, right? And so we need to find these hotspots of diversity. And the only way to do that is to know how many species are in these various regions. So like the Coral Triangle in the West Pacific, we know a lot of the diversity there. We know it's a very highly diverse place. And so we can say, okay, well, let's funnel resources. Let's target that area as an engine of biodiversity. Let's target that area as a place that generates diversity and protect that, right? Rather than looking at, you know, maybe some places like, I mean, it's hard to say, but at this point we have to prioritize. We can't put an umbrella over all of it. And so finding the diversity, the hotspots of diversity, sort of taxonomy and phylogeny is really important to know what's there so we can figure out where the bulk of it is, and kind of funnel our resources accordingly.
Cool. So you had mentioned that it's something, you know, thousands of species are described every year. And I guess I have two questions. So one, since it's the Zoological Commission of Zoological Nomenclature, do they also deal with plants, or are plants a different?
The plant people have different stuff. So the plant people one, they use a lot of Latin names, but they have different rules about how you have to contextualize them. And I'm not very good at that. My algae friends are very into it because they describe a lot of algal species, but it's different. It's not the same. And they have different rules, but like the zoological nomenclature covers all of the animals. So like insects, hippopotamuses, all of that go in that book. And the plant people have a different jam entirely.
So out of the thousands of animals species that are described every year, first of all, I guess I'm wondering, like, how many do we actually hear about, and what are the majority in terms of the type of animal that they are? I mean, we don't hear about thousands of, you know, mammals being described every year.
No, one of the things that I think plays into this question, you don't hear a lot of mammals being new mammal species being described, but you know what, mammals are very well studied. Terrestrial systems and the diversity of terrestrial systems is very well studied. Because we are terrestrial mammals, it's easy for us to get to place that other terrestrial mammals live. So I would say most of the new species descriptions that you see are marine. Most of them are deep sea. Just because everybody's very fascinated by that right now. And so there's a lot of funding that's going towards like submersible expeditions and stuff like that. But reefs are really important, and I think they're being kind of neglected right now. But most of the stuff you see is marine, just because there aren't a lot of hippopotamuses out there that we don't know about.
Right. Well, I mean, I remember every now and then there'll be something about like a primate species maybe in the rainforest, or I remember a few years ago there was a new mammal species in the South American rainforest, the Olingo, which was different from the kinkajou, and that was like a huge, oh my gosh, we have a new mammal species, and it was such a big deal. So then to hear, well, there are thousands every year, but we don't hear about these thousands.
There's a lot of insects, birds. We get birds, but a lot of marine invertebrates. Just because they've been pretty understudied. Just because the ocean's real hard. It's really hostile. We are not supposed to be there. It does not want us there. And so it's hard for us to study that.
Right. I mean, it's a very different, in many ways, it's a very different search, right? Because when it's terrestrial, first of all, the light aspect to begin with, being able to see things, but also just the vertical nature of a terrestrial, if you know it's on the ground, it's in vegetation, or it possibly can fly. But when you talk about a water column, that's the whole water column. So there's just so much more space.
All of the problems that we have in outer space, we have in the ocean. We can't breathe. There's pressure issues. There's no light. Well, that's not true in space. Never mind, planetarium lady. But you know, it's not a place we're supposed to be, and there are other organisms that are actively trying to kill us.
Good point.
So
it makes a difference.
It's hard and it's expensive. So.
Cool. So we had just a comment. Thank you for a great presentation and explanation of how taxonomy works. And I think certainly I have thought, and I appreciate that you suggested that I would get to name a species in 2021, because that will never happen. But it's nice to dream about naming a species and describing it and being the first to see something. But I think people don't always realize, first of all, the whole process that you have to go through, all of the work to make sure that what you're describing is in fact totally different, and also the decisions that you make on how you're going to say these are the reasons that it's different. You know, I don't think people always recognize that, and certainly when we hear about explorers or naturalists in the 1800s going someplace that they haven't been before, and oh, that's different and that's different, you know, it sounds like, well, you just look at it and you know, but obviously there's a lot more going on there.
Well, it's interesting that you say that. So there's a lot of artistry to it, and you have to have a deep knowledge of the other crabs to know if your crab is different, right? Because you don't know necessarily what's important to which group of crabs, right? Like in some groups of crabs, the pattern of spines on the claw is really important to differentiating the species. In other types of crabs, it's not. So you have to know that. And then as you're saying, like, it's not enough to just know because you looked at it. I had Yo-Yo, the little Indonesian lady, she told me something that one of her teachers used to tell her. And she said that Pat had told her one time, I can't figure out, but I know it with my heart. And sometimes you do look at it and you know, but because it's like filtering through your brain, you don't know why. But you do know because you've looked at so many. It's intuitive that you don't know why you know. And then you run the genetics and it confirms it, and then you're like, okay, I got to figure it out.
Right.
Got to figure out what's in there.
What's going on?
Yeah. I know it with my heart. And it's like, that's not science.
Well, it kind of reminds me of, I had an ornithology professor, and I was saying, so I saw this bird and this is what I saw, and he said, well, what did the tail look like? And I was like, I wasn't looking at the tail, I was looking at this. And so then the next week I went and I said, okay, so the tail looks like this. And he's like, but what did the wings look like? And I said, but you told me to look at the tail, so that's what I did. And then so the next week I was like, okay, so the wings look like that. And he said, but the head. I was like, I wasn't looking at the head, I was looking at these other things. And it wasn't until I went in there one time and I was like, the tail looked like this, the head looked like this, the wings looked like this, it was sitting in this tree, this is the noise it made, and this was the other bird near it. And he was like, now you figured it out. So I saw that whole bit of really paying attention.
Yeah, and that goes back to the idea that one character doesn't make a species. Right. Like even those finches of Darwin's, like to us looking at the pictures, it's really obvious and all of that, but it's like, no, the beak has to be shaped like this, and the tail, you know, and it has to have, and then you can call it.
Right.
You know, it's not just like one thing. And some of these hermit crabs, like, it's a struggle sometimes because it's like, it's a different color. And so that's when the genetics is really important, because you can get the different color and the genes say so.
Right. Thank goodness. Just imagine before we had those opportunities. Okay, I want to thank you so much. This was great. I don't know if anyone has any other questions or comments that they want to add. But if you're like me and you tend to think about questions afterward and you wish you'd said something, you will all be getting an email from me tomorrow thanking you and also asking for questions and comments. So you can respond back to me with anything you wished you had asked Cate, and I will pass it along to her. But I want to thank you all again for joining us. I hope you have a wonderful weekend. And I want to thank you, Cate, as well. This isn't a topic that I think we normally hear about, but it's certainly helpful, I think, to get an expert's perspective and also to recognize that this is a place where people are very passionate.
Yeah,
even if we're
like
not always
we
aware of
get
it.
kind of on our soapbox about it, you know? We get like, I get like misty-eyed when I think about Linnaeus and like the legacy and like, you know, maintaining that sort of craft of it. I get like misty, you know?
There we go. That's fantastic. Okay, so thank you, everyone. What I'm going to do is I'm simply going to end the meeting because that's kind of the only way that I know how to do this part. But I want to thank everyone again. This recording will be available so that if you think there's anyone else that might be interested in this, we'll share it in our blog and stuff like that so that everyone can see it. But I hope everyone has a wonderful weekend. If you are up in New Hampshire, I know it's supposed to get warmer and warmer and warmer. So hopefully we're ready for that. And then just kind of looking ahead to next month, we've got a woman who had been, and she still consults as a mitigation paleontologist, talking about her work and what it means to do that, and also what it means to be in New Hampshire where there really aren't any fossils. So look for information on that and other stuff that we've got going on. But I hope to see you all soon at some point when it is safe to do so. So good night, everyone.